Hello and welcome to the exciting new field of epitranscriptomics. So what is epitranscriptomics? For DNA genome, we have DNA and chromatin modifications that make up the epigenome to program gene expression. Likewise, RNAs in the transcriptome are specifically modified by many chemical groups to form epitranscriptome. There are regulatory chemical modifications on RNAs in the transcriptome that are installed at defined sites, that are evolutionarily conserved, that are inducible, reversible dynamic regulation. They're not about RNA sequences themselves. They modulate genetic information flow and functions. Similarly, proteins in the proteome are also modified with chemical groups such as phosphate group by kinases, methyl group by methyltransferases, and acetyl group by acetyltransferases. When we're talking about epitranscriptomics, let's first focus on the most prominent M6A modification on mRNAs. M6A is a methyl group modification at the 6th position of the purine ring. M6A modification occurs most heavily on poly-A+ mRNAs, compared with non-polyadenylated RNAs on the M6A immunoblot. M6A is widely distributed across all tissues, although the abundances vary, with brain being the highest. M6A is developmentally regulated, as we can see the dramatic level changes from early embryo to adult. Epitranscriptomic modifications are fundamentally important in biology. For example, epitranscriptomic modifications mark rapid transcriptome turnover during development and cell differentiation. The developmental programs require tiny switching of one cellular transcriptome to another to bring about phenotypic changes and definition of cell type and lineage. Not only the master transcription factors, or TFs, must activate and produce particular new pools of mRNAs, but also the old transcriptome must be swapped out. For example, during embryonic development, the high levels of maternal mRNAs accumulated in the eggs are rapidly replaced by zygotic mRNAs upon fertilization. M6A methylations of mRNAs is a mechanism that marks this rapid maternal to zygotic transcriptome turnover. On mRNAs, on average, there are about 3 to 5 M6A sites per 1 kilobase length of mRNAs. These modifications are enriched around the stop codon for mRNA or last exons in lincRNAs. The M6A location harbors many mRNA regulatory features, such as splicing, microRNA binding, translation, and stability. M6A peak have motif of DRACH. M6A is a reversible modification. M6A modification is installed on the RNAs by writers such as metal-3, metal-4, and metal-14. The modification is read by readers such as translation initiation factors, EIFs, splicing factors such as HNRMPs, RNA microprocessors such as DGCR8, and RNA binding proteins such as YTH family members. The M6A modification is erasable by dioxygenase such as AlkBH5 and FTO, first being hydroxylated into hydroxy M6A and then carboxylated into formyl 6A, and finally decarboxylated back into adenine A. Epitranscriptomic modifications are not limited to coding mRNAs. Other classes of non-coding RNAs are also modified by epitranscriptomic marks. For example, non-coding RNAs or lincRNAs are similarly modified by CAP, M6A, M5C, and pseudouridine as mRNA. Circular RNAs can be internally modified by M6A that contributes to their catalyst translation. Mid-size non-coding RNAs, that is, RNA sizes less than 200 nucleotides long, but greater than 6 nucleotides, such as small nuclear RNAs or snRNAs, small nuclear RNAs or SNO-RNAs, ribosomal RNAs or rRNAs are modified by M6A and other modifications. And the tRNAs that are most famously and heavily decorated by close to 100 chemical modifications, to the extent we have to dedicate tRNA modification field of study. So what are the molecular and biological functions of M6A modification? In the nucleus, M6A modification in the transcript is read by YTH-DC1 or cytidine arginine splicing factor, or SRSF2, which determines the splicing of the intron and alternative splicing. M6A modification also causes RNA stem loop fold structural change. This M6A switch is read by HNRMP-C and HNRMP-G readers. M6A modification is also read by YTH-DC2 readers to regulate the RNA nuclear to cytoplasmic export. Newly transcribed RNAs are vulnerable to nuclease exosomes, which is different from the extracellular exosomes, even they have the same name. M6A modified transcript can be bound to IGF-2BP and get stabilized. In female cells, M6A in X chromosome inactivation RNA, XCIST, can initiate X chromosome condensation and inactivation. In cytoplasm, M6A can promote translation efficiency. mRNA translation is initiated by forming a non-covalent loop among the 5' end cap, the initiation factor complex, and poly-A binding protein PABP. M6A readers YTH-DF1 is part of the complex. Naturally, the poly-A tail and M6A near stop codon can ensure the initiation of protein translation on intact mRNA only, so that the translation resources are not wasted by the degraded mRNAs, or even potentially harmful competitive peptide fragments. Circular RNAs are covalently circularized RNAs that can have open reading frames. However, without the 5' end cap, circular RNAs do not translate proteins and are non-coding RNAs. Surprisingly, with internal M6A modification, the M6A can be read by YTH-DF3 reader and act as a cap structure to recruit and initiate capless protein translation. Another major function of M6A is to mark mRNA for decay. Once M6A is bound by YTH-DF2 reader, the mRNA is degraded by first shortening of the poly-A. M6A mark degradation is an important mechanism of rapid transcriptome turnover. So why do we study epitranscriptomics? This is because it is a very exciting new field of epitranscriptomics and its broad significance in basic biology and clinical research. With pervasive M6A involvement in biology, aberrant M6A modification is also associated with human diseases and cancer in particular. For example, M6A is associated with cancer stem cells, M6A-MYC in acute myeloid leukemia, M6A ADAM19 and FXM1 in glioblastoma multiforme, M6A analog, KLF4 and HIF in breast cancer. In cancer metastases, M6A microRNA 126 is involved in hepatocellular carcinoma, microRNA 125B in colorectal cancer, and M6A YAP in pancreatic cancer. In cancer cell proliferation, M6A SOX2 and M6A HBxIP are involved in liver and breast cancer cell proliferation. Now we have come to appreciate the exciting epitranscriptomics in biology and disease. For each transcript is a pool of unmodified and modified RNA molecules. For each transcript, there are two key measurements of M6A modification: the M6A modified RNA abundance and the percentage of modified RNA molecules in that transcript. The abundance and the percentage of modification numbers are both important in epitranscriptomic profiling. For each mRNA, the M6A and its readers confers different metabolic fates and functional roles of the modified and unmodified RNAs of the transcript. For example, the unmodified RNAs can be destined for protein translation, whereas M6A modified RNAs can be destined for RNA degradation. The effects of an RNA modification depends on both the molecular consequences and the percentage of the modification. For example, if the molecular consequence of the modification is splicing, even a small percentage of modified RNAs can significantly impact the biologic effect, as the alternatively spliced isoforms can form a separate, different new function. On the other hand, if the molecular consequence of mRNA modification is RNA decay, a small percentage of the modification is not going to have a big enough impact on the overall RNA level. In this case, a large percentage of the modification is needed to cause appreciable RNA level change. Therefore, the M6A effect is contributed by both the molecular mechanism and the percentage of modification. Although profiling M6A by MeRIP sequencing can measure the abundance of M6A RNA, profiling the percentage of modification has been a big challenge. The lack of modification stoichiometry has been scientists' long and top concern in the field, as the commentaries in Nature Reviews of Molecular Cell Biology and published in Science. The other challenges of M6A epitranscriptomic profiling is transcript-specific modification. A gene can produce several alternative spliced transcript isoforms with different configurations of modifications and different, sometimes opposing biological functions. Such a combinatorial epitranscriptomic mark can dictate its splicing, stability, localization, and translation status, thereby providing a precise cell-type specific context for regulation. Methylated RNA immunoprecipitation sequencing, or MeRIP sequencing, has been used for M6A profiling. Although current MeRIP sequencing technology can map M6A site in mRNAs and measure the abundance as the peak heights, it has many limitations. That is, it cannot profile the modification stoichiometry or the percentage of modification. It is also difficult to profile M6A modification to specific transcript isoform due to generally low sensitivity of transcript-specific profiling by RNA sequencing. Furthermore, MeRIP sequencing requires massive amounts of RNAs of greater than 100 micrograms to do the experiment, which is not practically available under many circumstances. The much better solution to these challenges is epitranscriptomic microarrays. In a typical workflow, the M6A immunoprecipitated RNA fraction is labeled with signing 5, red color, whereas unbound, unmodified RNA fraction is labeled with green signing 3 color. Both fractions are mixed together and hybridized with the same microarray. The intensities in the two color channels can accurately calculate not only the abundance of the modified RNA, but also the percentage of RNA modification. Importantly, the microarray probes are designed to each transcript-specific splice junction, or unique exon sequence, which unambiguously and precisely measure M6A modification for each and every specific transcript isoform. So the microarray can tell what gene transcripts are modified, the percentage of RNA for each transcript, and the differential modification between the conditions and groups. It covers mRNAs and non-coding RNAs as well. It requires small RNA amounts, or around 1 microgram. And there's no need for poly-A selections or ribosomal RNA removal as required by MeRIP sequencing, or simultaneously on one array. Here is a table to recap why use epitranscriptomic microarrays or MeRIP sequencing for the current needs: RNA type coverage, transcript isoform specific profiling, starting RNA amount requirement, mRNA enrichment or RNA removal, and RNA integrity demand. Here are the specifications of Arraystar Epitranscriptomic Microarray contents. We have epitranscriptomic microarrays for human, mouse, and rat species, covering mRNA, lincRNA, and mid-sized non-coding RNA classes. We have also circular RNA epitranscriptomic microarrays that target specific circular junctions for the most sensitive, accurate circular RNA M6 modification profiling, not nearly possible by MeRIP circular RNA sequencing. The epitranscriptomic microarray is annotated and analyzed with M6A modification level, which is the percentage of M6A modification, for example, 45% or 3%. The M6A RNA abundance for each RNA transcript is calculated to provide these two key measurements of M6A modification stoichiometry and M6A RNA abundance. The differentially modified mRNAs in comparison between the groups or conditions are collected for gene ontological functional enrichment analysis and biological pathway analysis to gain functional insight to M6A impact on biology and disease. M6A profiles are hierarchically clustered and presented on the heat maps. With the most sensitive, accurate quantitative profiling data, the opportunity of epitranscriptomics research is now wide open. Thank you.